Cell structure. This is your second lab this week, week 3. Most of this should be review from previous lectures, but there will be a little bit of new information. Okay, so I'll try to go quickly. So the cell theory, as you recall, says that all living organisms are composed of cells. Cells are the structural and organizational unit of life. And with that, they are the smallest unit of life. And all cells come from pre-existing cells. Remember that this was the work of many scientists over time, improving the resolution of microscopes, examining more types of samples, learning about what microbes are, that they exist. But we do credit a few scientists in particular when it comes to cell theory. So you have Sheldon, a botanist. He found cells in all plants that he examined. So he, said that all plants are made up of cells. And then you have Schwann, who came to the same discovery with animal tissues. And this might not sound impressive to you, but you have to get really thin slices of these tissues, plants and animal tissues, to be able to examine them under the microscope. And you have to have a pretty good microscope. So it's actually pretty impressive. So they both saw, like, okay, most living things that we know about are either plants or animals, and they all have cells, right? And so they also, that came together with Virchow, remember, who said that that all cells come from pre-existing cells, but he didn't have proof of that. But together, all the work of all the different scientists, we came up with the cell theory. Okay. And so really this work started back in 1665 with Robert Hooke and also Van Leeuwenhoek, when they came up with finding cells in microorganisms for the very first time. Okay, eukaryotes and prokaryotes. So you have to remember the difference. You also need to know each of the parts of each of these cell types and their function, okay? So you can go back to chapter 4 and review that in lecture. But in brief, remember that all living cells will share the 5 basic components. Every single cell has to have a plasma membrane. It's going to have cytoplasm. It's going to have genetic material called DNA. And it's going to have ribosomes enable the cell to make proteins, remember. And then, of course, some type of cytoskeleton, whether that be very simple, like you see with prokaryotes, or more advanced, like you see with eukaryotes. Okay, so those are the basic components of all cells. Okay, so prokaryotic cells means no nucleus and no membrane-bound organelles. So this includes the 2 domains of life called bacteria. Sometimes you'll see it with a EU in front for eubacteria. And then archaea bacteria, sometimes just called archaea. So those are the 2 domains. The third domain, of course, being eukaryotes. So these 2 domains, bacteria and archaea, are prokaryotic. So they are single-celled organisms, no nucleus, no organelles. You will find the small ribosome type called 70S ribosome. The 70S ribosome is composed of 2 parts, the large and small subunits. The large subunit being 50S and the small subunit being 30S. Remember, these are not additive. The S stands for the unit of centrifugation. Okay, so these are centrifugation units, not mass. They are related to mass, but they are not directly mass, and that's why these are not additive. Okay, circular DNA in the cytoplasm region, called the nucleoid. And then bacteria can also have small circular DNA units called plasmids, and plasmids are going to give extra genes that are not required for everyday life, but can help give special properties like antibiotic resistance. Remember that plasmids are shared between cells through the pilus. Eukaryotes are more complex cells. These can include unicellular and multicellular organisms. Okay, so animals, plants, protists, and fungi are kingdoms. There are many more types of kingdoms within the domain Eukarya, but these are the ones that we focus on. So these all are going to have specialized organelles, a little bit of variation depending on the type, but for the most part, they all will have a nucleus, an endoplasmic reticulum, a Golgi apparatus, mitochondria, and lysosomes. Okay, they also will have the larger ribosome type made of the 60S and 40S subunit, and so this ribosome is called the 80S ribosome. So again, not additive, but you do have a larger large subunit and a larger small subunit. Then remember that viruses are excluded from this hierarchy nomenclature of life, right? Because viruses are not living. Viruses have to get inside of a living cell in order to replicate. Viruses do not have a metabolism, right? There's a lot of reasons why viruses are not considered living, but they are infectious. Okay, so that's a big difference. Remember that the cell membrane is made up of a lipid bilayer, and specifically a phospholipid bilayer. They are selectively permeable. We learned about the different types of transport across the membrane, right? Active requires ATP. It is like a pumping across the membrane, going from an area of low concentration to an area of high concentration. So it's going against the concentration gradient. That is in contrast to the passive types of transport, which go down the concentration gradient from high to low. Passive transport can include simple diffusion or facilitated. 0 ATP is used because it's going down the concentration gradient. We also learned that there's all sorts of receptors on the cell surface for hormones and other signals to bind to. We learned that the fatty acid tails of the phospholipid bilayer hide inside because they are hydrophobic, right? And that the phosphoheads are hydrophilic and they face the aqueous solution both on the outside and the inside of the cell. And that the membrane itself behaves in a fluid mosaic fashion, right? It's not static. Things move around. The membrane moves a little, and the parts within the membrane can move around as well. And then these links you've seen before, but go ahead and watch them again just to refresh and study. And remember that cholesterol is only found in animal cells. Okay, other cells will have more simple versions, but the function is to provide more stability and allow these phospholipids to kind of stack nicely, tight, neat, in a neat fashion. Okay. Cell walls depend on the type of cell. Not all cells have a cell wall. But the different groups of organisms have their own characteristic cell wall. So remember, bacteria, most bacteria have a cell wall. We saw just the one exception of mycoplasm. Everything else has a cell wall, and those cell walls will always be made up of peptidoglycan. On the other hand, we saw that plant cells will have, cellulose, fungi cells will have chitin, and archaea cells will have their own, what we call pseudomarin. It's a different type of carbohydrate. Of course, when it comes to animals, animal cells don't have cell walls. Okay. A cell wall is a strong structure located just outside of the plasma membrane, and it gives extra support, strength, right? It also helps to control what goes in and out a little bit. Okay. And we started talking about the difference between two major groups of bacteria called gram-positive and gram-negative. Here's a reminder here. Gram-positive have that really thick outer cell wall, and then just the plasma membrane below that, whereas gram-negative have that outer membrane and then a thin cell wall and then the plasma membrane. And so these will behave differently with their environments, right? Having a phospholipid membrane on the outside versus having the cell wall. Remember that only gram-negative and all gram-negative cells will have that LPS, lipopolysaccharide, in their outer membrane, okay? So bacterial outer cell structures, right? The cell wall, as we talked about, gives its shape, semi-rigid, flagella and other structures like fimbriae attach to the cell wall. This helps maintain the separate environment between the outside and the inside, in addition to the plasma membrane. And all bacterial cell walls are made up of peptidoglycan. Composition is used to differentiate types of bacteria, as I mentioned, gram-positive or gram-negative. If a bacterial cell has a capsule, that's going to provide extra protection. It's it's a different type of carbohydrate layer that hides those surface proteins on the cell wall. And so this is why we say that it's like a cloak of invisibility, right? Our immune system is not going to really be able to detect it because the capsule is covering up those surface antigens. The cell wall is particularly useful in resisting osmotic pressure, right? Because the concentration of substances inside the cell is much greater than the concentration of those outside of the cell. So water is going to want to rush into the cell, and the cell wall prevents that from happening. All right. Remember that peptidoglycan is made up of two monosaccharides called NAG and NAM, and is all of that is held together through the polypeptide chains and cross bridges. Gram-positive has a teichoic acid that helps give the cell wall, extra support. Teichoic acid is an alcohol and a phosphate, and you can have it be strictly located within the wall, or it can transverse from the membrane to the wall. Okay. And gram-negative, as I mentioned, has LPS, lipopolysaccharide, in its outer membrane. And we talked quite a bit about that. We'll talk a little bit more about it now. So remember that gram-negative bacteria stain red. Sometimes you'll see pink, but it's really more red than it is pink. And that's because the alcohol step in the staining process will wash away the crystal violet, and then the counter stain is all that will be seen, right? Review that from chapter 4 lecture. You can review it in your textbook in chapter 3. And the space between the membrane and the wall and the inner membrane is called the periplasm. And having this outer membrane does offer some protection against certain antibiotics. Remember we talked about LPS having these 3 layers. The outer part that sticks up is called the lipid A. I'm sorry, the outer portion that sticks out is called the O antigen. And then the part that's embedded within the lipid is called lipid A. And when this becomes free from the cell, and then the host becomes exposed to it, this can act as a toxin. So we call it an endotoxin because it's usually inside of the bacterial cell. But when it does become free, then it can act as a toxin. It can cause fever, diarrhea, and can even lead to endotoxic shock, or what is also called septic shock. And so a little bit about shock, because bacteria have there's 3 main categories of shock when it comes to bacteria. And you should really make sure to save this slide for when we go over the survey of bacteria, because this will become a useful guide for you. But any shock is a generic medical term that means your vital organs are not getting enough blood flow, and that means that they are at risk of shutting down, and then of course that puts you at risk of death. Okay. Toxins, certain toxins of bacteria are able to overstimulate the immune system and cause shock. Okay, so we have toxic shock syndrome, which you've probably heard. It became very popular in association with tampon use, although nowadays it's rarely caused by that, and is mostly caused by like poor food hygiene, for example. Staphylococcus aureus can cause a whole range of diseases depending on where it gains entry into the body. It is a gram-positive bacteria, which it's the main bacteria responsible for toxic shock syndrome. We have another type of gram-positive bacteria called Staphylococcus pyogenes, which is responsible for streptococcus toxic shock syndrome, STTS rather. And this has a special ability, which we call beta hemolytic. It has hemolysins, which can completely break down red blood cells, but it doesn't only break down red blood cells. It can, turns out, it can break down pretty much any type of cell. You may be familiar with this when it comes to strep throat. You get that incredibly red, incredibly sore, swollen throat, and that's Streptococcus pyogenes. Now, what did I want to tell you? Okay, so in addition to this hemolysin, there are other exotoxins that can lead to shock. Okay, so if Streptococcus pyogenes gets into the deep layers of the skin, or if it enters the bloodstream, that's when you are likely to develop the shock syndrome. So, you don't need to worry that if you have strep throat that you're going to, have shock. That's 2 completely separate things. But Streptococcus pyogenes is the same organism. Also, we always treat strep throat for a few reasons, and we'll talk about that when we do our overview of bacteria. Okay, the third type of bacterial shock is the one caused by LPS. It's septic shock, specifically septic shock syndrome. And this is the gram-negative bacteria, and it's from LPS, and it's an endotoxin. Okay, so you need to know the differences there. Okay, gram-positive, gram-negative. Remember, gram-positive purple, gram-negative red. They can be different shapes and different arrangements, but all gram-positive have that thick cell wall, and all gram-negative have the outer membrane, thin cell wall, and plasma membrane. Okay, so remember the structure of these cells and why they stain the way that they stain. We talked about bacterial morphology in lecture. So just remember that if it's round, it's cocci. If it's rod-shaped, it's bacilli, and others are like spiral. So you have your spirochetes, your Spirilia, and your Vibrio. And if it's in a chain, it's strepto. If it's in a cluster, it's staphylo. Some bacteria are able to move around, and that will be typically due to the presence of one or more flagella. If it's highly motile, it will be capable of swarming. Okay, so go ahead and review bacterial motility, the names of the arrangements of flagella, and how they move, right? All of the flagella have to move together in counterclockwise rotation in order for it to move. And if one or more of them starts to move in the clockwise direction, it'll cause them to spread apart, and it'll cause the cell to then tumble. And so we say that if there's an attractant that it's running towards, you'll see kind of a biased random walk where there are fewer tumbles and longer periods of runs, right? Which we saw here. So make sure you know your term chemotaxis and you understand how they move towards a stimulant or away from a harmful stimulant. We learned that fimbriae are like short fingers. They are short, fine hair-like projections that are good for attachment, for colony formation. They are not used for motility. We learned about pili, which could be one or more pili on a cell, typically used for the transfer of plasmids, and that's through a process called conjugation. But pili can be used to grab a hold of a surface and pull the cell towards it, and it can release and repeat this multiple times, and that can lead to like a twitching type motility. So remember, fimbriae, never motility, pili, sometimes. And then we learned about axial filaments, which are also a motility structure that some spirochetes, all spirochetes have this, and it's anchored at one end. It wraps around the cell, and then that whole structure is covered in like a thin covering called a sheath. Okay, and other bacterial components, remember we talked about the circular chromosome of DNA kept in a region called the nucleoid. This contains all the regular genes for living, so we call these housekeeping genes. If it has a plasmid, it's a small extra chromosomal piece of DNA for special advantages like antibiotic resistance. All bacterial cells have the 70S ribosome. This is how they make their proteins, the part of protein synthesis called translation. Inclusion bodies are storage vesicles, typically of nutrients, and you will look at the different types of inclusion bodies in the simulation. We briefly went over them in lecture. You read about them in your book, and then you'll see them again in the simulation. And that is it. I went really fast because, like I said, most of it was review. Make sure you read everything in the simulation. You also have a handout. It looks like Labster has started to make these like lab manuals to go with the labs. So if they are available, I will provide them to you. You also have the theory pages to take your screenshots of the theory pages to help you study as well. All right, that's it. Bye.